a bison approaching a violently erupting geyser. Then a bright yellow mat of thermophiles, followed by images of wildlife in quick succession.
Welcome to Yellowstone, land of geysers, hot springs, dazzling colors, wolves, bears, and bison. You probably came to Yellowstone to enjoy these places and animals.
Panoramic view of a large hydrothermal feature. A small geyser erupting next to a bright green stream. Columns of water burst out of the ground, rippling out in a circle around the geyser. Then plumes of steam rising up into the sky.
What you may not know, however, is that there are actually millions of organisms that are helping put on this show. They are called thermophiles.
A large gooey bright yellow mass of microbes.
Yes, that is them. They are everywhere in the park.
A close-up of underwater thermophiles. They are a dull yellow and form mats made up of long, thick filaments.
What exactly is a thermophile?
Annie Carlson, Research Coordinator at Yellowstone National Park.
A thermophile is a heat-loving organism, and typically we're talking about quite high temperatures, more than 100 degrees Fahrenheit.
Light yellow and brown fuzz covering the rocky formations around a body of water.
Dr. Bill Inskeep, Professor of Geomicrobiology at Montana State University.
And we're not talking about an organism that can simply tolerate high temperature. That wouldn't be considered a thermophile. We're talking about an organism that has its optimum growth temperature at a high temperature.
Countless tiny yellow bumps protruding from the water, then a large mat of dark yellow sponge-like structures.
To give you a point of reference, we could be considered mesophiles. We have an optimum temperature from 20 to 35 degrees Celsius, and we can even tolerate up to 40 degrees. Thermophiles prefer it a bit hotter, from 40 to 80 degrees, and Yellowstone is even home to hyperthermophiles that live in temperatures above 80 degrees.
Rocky steps covered in a fuzzy brown substance traverse a snowy plain, while visitors stroll on a boardwalk in the distance.
The thermophiles of Yellowstone come from all three domains of life: archaea, bacteria, and eukarya.
Panoramic view of a wetland. A small stream traversing the area is painted dark brown due to the presence of underwater thermophilic communities. Then, an animation showcasing the various distinct types of archaea, bacteria, and eukarya. Categories of bacteria include proteobacteria, chloroblasts, cyanobacteria, flavobacteria, thermotoga, aquifex, Thermatosulfobacterium, gram-non-sulfur bacteria, gram-positive bacteria, and mitochondrion bacteria. Archaea include Corarchaeata, Thaumarchaeata, Euryarchaeata, Cronarchaeata, and Nanoarchaeata. Eukarya include fungi, plantae, ciliates, flagellates, microsporidia, diplomonads, trichomonads, entomoebae, myxomycota, and animalia.
Archaea are single-celled organisms, and they can often be found in the hottest areas of thermal features, with temperatures in the neighborhood of 100 degrees Celsius. Bacteria, which are also single-celled organisms, prefer environments with slightly lower temperatures, around 85 degrees. Eukarya, which can be multicellular, include algae, fungi, plants, animals, and even us. Thermophilic eukaryotes live in even cooler areas and aren't found at temperatures much past 60 degrees.
Large clouds of steam rise up from a white terrace.
One thing you might be wondering is why thermophiles can survive such high temperatures when we can't.
Thermophiles, their cells are really amazing. They can survive at high temperatures because their genes and their proteins are different than ours. Human cells, our proteins would denature at high temperatures.
Long strips of brown and light green along the length of a tall hill. Then animation. A red thermometer showing a temperature of 176 degrees Fahrenheit. Next to it, two rocky square blocks labeled humans and thermophiles respectively.
Which means that as temperature rises, our proteins fall apart.
A flaming sphere crushes into the block labeled humans and breaks it into tiny pieces. Next, the sphere attacks the thermophile block, but it is knocked back, leaving the block unscathed.
On the other hand, the proteins inside the cells of thermophiles stay rock solid and are unaffected by the heat.
A vast land covered in hues of yellow and brown encircle a bubbling pool. Then, an overhead view of a multicolored hot spring. Its bright yellow edges give way to light green, leading up to a deep blue center. Next, steam rising out of a long yellow river.
Yellowstone is home to countless species of thermophiles, but they don't all live in the same areas of the park. As it turns out, water temperature and chemistry are the two most important factors in determining where thermophiles live?
Probably the most important is temperature. Some organisms prefer 80 to 90, some prefer 70 to 80. So we can find a distribution of different thermophiles just based on temperature. Of course, chemistry plays a secondary but extremely important role in the distribution of different thermophiles.
A sequence of scenes showing patches of land entirely covered in yellow, brown, or white mats, emitting plumes of steam in the air.
For example, if you have a high iron system, we might have organisms that are oxidizing iron. If we have high sulfide, we might be oxidizing sulfur for the energy. And therefore the chemistry combined with the temperature all of a sudden starts to control the distribution of what organisms are present.
Overhead view of a bubbling pool. Its deep blue center is encircled by yellow and white clusters of growth.
Besides temperature, the water's pH is another important parameter that affects where different species of thermophiles occur.
A rapid stream rushing down a bright yellow slope, then an animation of the pH scale.
So what exactly is pH?
pH is a measure of the acidity of water. So perfect distilled water is neutral. It's a 7 on the pH scale, whereas moving to higher levels of pH, you get more basic. Things like drain cleaner are about a 14 on the pH scale, so very basic, whereas things like battery acid are very, very acidic, would be about a 1 on the pH scale. We have features in Yellowstone that are 1 or below, very, very acidic features. We also have some that are quite basic, and we have thermophiles that live in all of these environments.
Sulfur Cauldron has a pH of 1.7. The Little Whirligig Geyser has a pH of 3.3, while that of the Morning Glory Pool is 5.8. The Grand Prismatic Spring has a pH of 8.3, while the pH of the Grand Geyser is 9.5.
As you travel around Yellowstone, you will encounter several different thermal areas, and they're all unique.
A large terrace made up of yellow rocky steps, forming small pools and streams. Half of the terrace is covered in snow.
Places like Mammoth Hot Springs, where we are now, have a very unique bedrock, a limestone underneath us. So the mineral that comes up to the surface is called calcium carbonate, and it forms a rock called travertine.
Water trickling down a textured light yellow surface, followed by a bird's eye view of a bright green stream crossing a gray flat land.
The water itself here is neutral water that is not boiling, it's not too hot. Whereas other areas like Norris Geyser Basin, we would call acid sulfate areas, where the water itself is very hot, but also the water and the gas coming from underground carry hydrogen sulfide, and this ultimately becomes sulfuric acid. So the springs may be very acidic, and you have different microbes that will thrive there.
Panoramic view of a wide river. Steam rises up from the gray riverbanks, then water streaming down into the river, leaving behind streaks of yellow on the white rock.
And then you have other areas, like the Upper Geyser Basin, for example, we would call neutral chloride, where the water, it's more neutral, maybe even a little bit basic. It can be quite hot, though, so different microbes will thrive in those environments.
Overhead view of a wide, shallow stream with patches of dark brown visible on the riverbed, then streaks of color on a large rock, followed by a multicolored spring.
As you're out exploring all these different environments, you'll see many different species of thermophiles. But how can you tell them apart? Along with water temperature and chemistry, it just so happens that all those beautiful colors you see are important clues.
Side view of the Grand Prismatic Pool. An animated sign pointing to the bright blue center of the pool reads, "Center of pool, no thermophiles, 87 degrees Celsius or 189 degrees Fahrenheit." Scientists are still researching if thermophiles live at the height temperatures in the center of hot springs.
Color of thermal systems in Yellowstone is a wonderful diagnostic tool. If you look at Grand Prismatic and start from the middle of the pool, you can tell there's no organisms there because it's only the blue color of the water. And a lot of that blue color in the water comes from dissolved silica. And dissolved silica is very high in hot water systems.
A text box pointing to the yellow inner ring of the pool reads, Senecio-Caucasus Zone, 67 to 75 degrees Celsius or 153 to 167 degrees Fahrenheit.
Then as you work cooler, you get to a band of yellow color that by definition we know that it's probably no hotter than 75 centigrade right at that point because that's where photosynthesis can start.
A text box pointing to the outer orange ring of the pool reads, Senecococcus chloroflexus zone, 60 to 68 degrees Celsius or 140 to 155 degrees Fahrenheit.
And the organisms then that live in the yellow band are a little different than the organisms that live in the orange band, and they're expressing different pigments to capture light energy.
A text box pointing to the darker brown banks of the pool reads, Calithric zone, 30 to 45 degrees Celsius or 85 to 140 degrees Fahrenheit.
So you can have a lot of different species of thermophiles living right next to each other in these beautiful banded patterns based on their optimum temperature range.
Sweeping view of large terrace rock formations. Streaks of yellow and light brown traverse their bright white surface.
In fact, one runoff channel, such as this one at Mammoth's Mound Spring, can have hundreds of different species of thermophiles.
A sequence of shots showing various types of thick, spongy mats made up of bright yellow and light brown filaments. Then various shots in quick succession. Flies, a spider, and a bird hunting for prey across the thermophilic zones.
What's incredible is you can't see each living cell, but when they live all together like that, they create these beautiful and very unique mats all around the thermal features. Thermophiles are the smallest building block of this ecosystem. Starting with the archaea and bacteria, you'll then have bigger and bigger organisms that will start to eat those cells. So you'll have some eukaryotes, which are protozoa, which will sort of swim around eating archaea, eating bacteria. And from there you'll have some ephedrid flies. You might have spiders be a predator of the insects. And then even birds will come in, like killdeer will start to come in to eat the insects.
Bird's eye view of a thermophilic area. Streaks of bright brown create a picture of web-like veins across a flat land.
While thermophiles are stunning, unique, and fascinating to see in Yellowstone's thermal areas, they are also a subject of intense scientific research, and they hold a lot of potential.
Two men discussing in front of a computer. Then a large metal apparatus, followed by a time-lapse of the starry sky at night, with plumes of steam constantly rising out of the ground, and a view of a hydrothermal feature entirely hidden by thick fog.
Life may have evolved in high temperatures. If life started at high temperature, then we have a beautiful place to study the possibility of those processes. Yellowstone itself is interesting because it has more geothermal systems than the rest of the globe combined. So Yellowstone is an exciting area to study high temperature biology and may give us clues of the origin of life.
A bubbling, raging mud pot.
And it is not just the origins of life here on Earth. NASA has sponsored research in Yellowstone because the hot environments found in the park might mimic environmental conditions on other planets. The thermophiles here could give us clues about the potential for life outside of Earth.
A scientist, wearing gloves, probes at a small circular metal device with a pair of pincers, then a large scientific instrument.
Studying the origins of life is clearly important, but that wasn't the only thing scientists were interested in when studying thermophiles.
One of the greatest discoveries that has come from thermophiles in Yellowstone can be traced back to Dr. Thomas Brock was working in the park in the 1960s. He isolated a bacterium that we call Thermus aquaticus, and a couple of decades later, a scientist named Carey Mullis isolated an enzyme called Taq polymerase from the microbe. Using Taq polymerase, Carey Mullis then invented a process that we call polymerase chain reaction, or PCR, that is now used in laboratories across the world to amplify strands of DNA. PCR is used in things like crime scene investigation. It's also used extensively in biomedical research, looking for ways to cure cancer.
And that's not all. PCR is both part of screening for genetic diseases and the early detection of infectious diseases, such as HIV.
An animation showing the various uses of the polymerase chain reaction. When used in the field of biomedical research, it can help detect genetic diseases such as trisomy 18, Fragile X, Down syndrome, cystic fibrosis, and sickle cell anemia. It can also detect infectious diseases such as tuberculosis, HIV, and pertussis.
It is also used to amplify DNA from the remains of our ancestors, helping shed light on the evolution of early humans.
A series of shots showing various types of thermophiles, bright green strands floating in a stream, then brown vein-like structures and bright yellow mats extending underwater.
It is amazing that a tiny microbe, discovered in Yellowstone, has had a big impact on our everyday lives. That is why it is important to preserve these thermophiles, because they may contain future scientific discoveries.
Visitors walking along a boardwalk traversing a light brown flat land, streaked with bright green and dark brown filaments.
Below the boardwalks are these vast microbial mats where all of the cells are living at this tiny microscopic level. So if you were to step onto the mat or put your hand in the water, you could impact these microbes.
Time-lapse of large crowds strolling along the winding boardwalk surrounding a large, hot basin.
There's not that many of these environments on the planet. They may contain interesting and rare life forms that we've never studied. If we don't protect these resources, it could be easily destroyed.
Panoramic view of vast dark brown wetlands. Thick billows of steam rise high into the air.
As you admire all the different thermophiles spread across Yellowstone, hopefully you have a greater appreciation for these tiny organisms and their ability to live in such extreme and inhospitable places.
A beautiful terrace with streaks of thermophiles painting the rocky steps in various hues of yellow, brown, and green. Then a river cascading down a hill of brown and yellow.
The iconic species of Yellowstone, like bears, wolves, and bison, are definitely worth seeing. But thermophiles might just be the real stars of the show. The sheer diversity of colors, textures, shapes, and patterns they bring to Yellowstone's thermal areas helps create an environment found nowhere else on Earth.
An overhead view of a multicolored hot spring with steam coming out of its banks.
Directed and produced by Hugo Sindler. Producers Bianca Klein and Dave Kruger. Featuring Dr. Bill Inskeep and Annie Carlson. Narration Sarah Lanier. Camera and sound Patrick Tormey, Sarah Lanier, and Chema Dominic. Original score Luke Sheeler. Production assistants Bianca Klein, Dave Kruger, and Janine Waller. Additional animations by DNA Helix, Wikimedia Commons, used with permission of Richard Wheeler. Special thanks to Tammy Blackford, Rachel Cudmore, Tony Purpura, Colette Campbell, Theo Lipford, Nate Reeders, Dr. Recep Avki, Nikki Jimenez, and Rick and Patty Sinsalar. Made possible by a donation from Canon USA Incorporated, through Yellowstone Forever. Produced as part of the MFA program in Science and Natural History Filmmaking in the SFP Montana State University Bozeman, sfp.montana.edy/science nature film.